In the next piece of the big picture, we'll be talking about how we get from DNA to RNA to protein. The DNA of a gene is a linear sequence of many nucleotides. It's going to need to be transcribed into RNA, and that will eventually be translated into a protein. So how does all of this happen? Different regions of chromosomes have different genes. And genes are sequences of DNA that encode a protein or a function. Different nitrogenous bases on the same region of homologous chromosomes can lead to different alleles. So for example, in the right here, we have a small difference in this gene. In one version, there's a CG pair, and in the other version, there's a TA pair. This would be an example of different alleles. One of these alleles might encode one color of eyes, like blue, and the other might encode another, like brown. If we want to express the eye color that these encode, we would need to transcribe the DNA into an RNA. This is called transcription because it is like making a copy or a transcript of something. Then we would want to convert this into the protein that it represents. This would be translation. In this case, we are moving from the nucleotide sequence of RNA to the amino acid sequence of a protein. It's like translating a language. In order to make a transcript, DNA helix has to be unzipped. RNA nucleotides have to line up, and the RNA polymerase has to join them along the strand of the DNA, following the base pair rule. RNA transcription has 3 steps: initiation, elongation, and termination. So let's talk about what is included in each of these steps. In order for RNA synthesis to begin, an RNA polymerase must find something called a promoter. A promoter is a region of DNA that tells the RNA polymerase that a gene starts here. It also gives information about where the gene should be expressed. We'll talk a lot more about promoters and other regulatory regions when we talk about how genes are regulated. Once the RNA polymerase has bound to the promoter, initiation can move forward. The next step is elongation. During elongation, RNA polymerase will read the DNA and will add free RNA nucleotides to make the RNA. This will continue until the RNA polymerase reaches the termination sequence. The terminator DNA or termination sequence tells the RNA polymerase that it has reached the end of the gene and therefore the RNA is complete. This is the termination step. If you're not comfortable with how this works, there's a lovely animation about it in the PowerPoint. Once an mRNA is produced, it's not quite complete. mRNAs contain multiple regions of information. These regions are often called introns and exons. Introns are regions of genes that do not code for any protein. And exons are the regions of genes that have the instructions to make a protein. Most of our DNA does not actually code for proteins, and most human genes have multiple introns in them. This means that there's information inside the mRNA that we don't need for translation. In addition to that, there is a cap and a tail, and these are important for helping the mRNA stay stable inside the cell and allow it to interact with the ribosome that's going to translate it. Before we can go through translation, we are going to need to cut out the introns, the regions of the gene that don't encode for the protein. And then we are going to use enzymes to join the ends together to create an mRNA that includes a cap, a tail, and only the part of the gene that actually encodes the protein that we want to translate. We'll talk a little more about why we have a process like that when we get to gene regulation. Once we have this mature mRNA, then we're going to have it leave the nucleus and enter either the cytoplasm or the endoplasmic reticulum, depending on where it needs to be translated. The kind of RNA that encodes amino acid is called a messenger RNA, or mRNA. It's called that because it contains a genetic message that's going to be used by the translation machinery. Translation requires converting nucleic acid language into polypeptide language.